name is Zepeda Rivera, and I'm a staff scientist in Dr. Christopher Johnston's laboratory at the Fred Hutchinson Cancer Center in Seattle, Washington. And I'm really excited to be here today to tell you about a snippet of the work that we have been doing to characterize novel bacterial species from the colorectal tumor niche in a highly collaborative project between our lab and the lab of Dr. Susan Bullman, also here at the Fred Hutch. So the colorectal cancer tumor microenvironment is a really complex collection of different cell types. So you've got malignant cells, and then you've got various invading and non-invading immune cells. However, an often underappreciated component of the tumor microenvironment is the tumor-associated microbiota. And this can refer to tumor-associated bacteria, viruses, and fungi, but we specifically are really interested in tumor-associated bacteria. Now, this tumor microenvironment is a largely unexplored niche, and the focus of the work I will tell you about today is to isolate, identify, and characterize tumor-associated bacterial species. What has been really surprising to us is that we've actually been isolating novel bacterial species from patient tumor samples. And you might ask, why does that matter? Why is it important that you're isolating novel bacterial species from patient samples? In addition to the fact that it's just neat to discover novel bacterial species that nobody else has identified or characterized before, the isolation of novel bacterial species from this highly specialized niche actually tells us a lot about the biology that is occurring in this environment. So when people refer to the tumor microbiota, in general, they're discussing stool samples from patients with colorectal cancer. And in human microbiome research, human stool, as well as the microbiome of other human body sites like the human oral cavity, have been extensively characterized through a myriad of approaches and techniques. In contrast to that, what we are studying is the microbiota that is physically on, in, and within the tumor tissue of these patients. So if we are identifying novel species from the tumor niche that have not been identified from a different human body site or a different human sample, then that species is likely specialized for the tumor niche. And we can study these bacteria to understand what these organisms do, and consequently, what those functions tell us about the biology that is occurring in the tumor microenvironment. So in the work I will be presenting to you today, we analyzed 4 tumor tissue samples from patients with colorectal cancer, and we identified 4 novel bacterial species across 3 of these samples. The 75% discovery rate supports the claim that the microbiota in the tumor microenvironment remains largely uncharacterized, and that likely there's a lot more species that are just waiting to be discovered. Due to the overabundance of human DNA, tissue-associated microbiomes are unfortunately not amenable to metagenomic analysis. And so instead, bacterial 16S rRNA sequencing is broadly used to characterize the microbial composition of these samples and get a very broad overview picture at either the species or the genus level of who is there. So as an example, here in the upper right, I'm showing you the % relative abundance on the y-axis for primary colorectal tumor samples along the x-axis. And you can see that along each column or along each patient sample, there's a different combination of colors, where each color represents a different bacterial genus in this case. Now, our goal is to functionally and mechanistically characterize the bacteria that are there in order to understand whether and how these microbes are impacting cancer initiation, progression, or even potentially patient response to treatment. But in order to do that, we require physical access to these isolates in order to then go to the lab and run experiments with them. And in order to gain that physical access, we've applied a multitude of technologies and techniques. The very first thing we did is we wanted to get an idea of what the microbial composition across these patient samples looked like. So on the left here, I'm showing you RNA-scope fish images of tissue sections from each of these patients. And in bright green, you can see the intratumoral bacteria. So again, these are bacterial cells that are physically on, in, and within these patient tumor samples. And you can hopefully appreciate that the bacteria are present across the tumor samples, and that there's areas of high bacterial abundance and areas of low bacterial abundance. When we performed 16S-based microbiome analysis of this primary patient tissue, we see that there is species heterogeneity across these 4 samples. So in the graph on the upper right, what I'm showing you is the % relative abundance on the y-axis across patient tissue samples on the x-axis. where colors within each column now represent a distinct bacterial species. At the very top, you see these white boxes, and those represent organisms that were found at less than 1% relative abundance that were all binned together into this other category. Now, if instead of doing microbiome analysis directly on the primary tissue, we first subject the bacteria to growing up in broth culture, and then we perform microbiome analysis, what we observe is that there's a drastic decrease in the species diversity. So on the lower left here, I'm showing you a graph of the alpha diversity on the y-axis for either the primary tissue microbial community or the microbial community from culturing in broth. And you can see that there's a drastic drop-off. So while we may be losing species diversity, what we are gaining by doing this is the ability to now detect low abundant species that were previously binned into that other white box at the top. So while 16S rRNA sequencing has the advantage of being able to bypass culturing, it is greatly limited to the species and the genus level resolution. So we're not able to get to strain level resolution with just the 16S rRNA gene. Additionally, identification of your bacteria is very dependent on the databases that you use and the inherent biases present in each of those databases. Because our goal is to functionally and mechanistically characterize these bacteria, and we require physical access to them in order to get there, the fact that 16S rRNA sequencing in and of itself does not get you physical access is one of the biggest drawbacks for us. However, we are able to apply other techniques in order to gain that physical access. For example, we can turn to traditional microbiology culturing approaches, where a sample, in this case a tumor tissue sample, is spread onto a Petri agar plate, and you can then pick colonies of interest and streak them onto fresh plates over and over again until you get a pure clonal population. By taking this approach, we've actually identified a novel Fusobacterium species from one of these patient samples, and we call this isolate SB21. And on the upper left here, I'm showing you what SB21 colonies look like on a Petri agar plate. So these cells form a distinct, vaguely round, somewhat opaque colonies. And when we take these cells and put them under a transmission electron microscope, we can see that individual cells have a spherical shape. And the spherical shape is actually very rare and unique for Fusobacterium species members. Most Fusobacterium species are these long, elongated rods. In fact, the genus is named for their fusiform shape. And so the spherical shape for SB21 was quite unique and neat. We could apply PacBio smart sequencing to obtain a complete and closed genome for this isolate that we could then use to perform comparative genomics using different bioinformatic tools, all of which indicated to us that SB21 is a novel Fusobacterium species. I'm showing you the results from one of these tools on the bottom left here, this case SNP phylogeny, which is a reference-free whole genome phylogeny approach. And you can see that F. sphaericum, which is highlighted here in blue, forms its own distinct branch away from all other known Fusobacterium species. Now, due to its spherical cellular morphology, we've proposed that this novel species be named Fusobacterium sphaericum. Now, the application of PacBio smart sequencing to obtain a complete and closed genome for a Fusobacterium species member is a huge advantage to us over Illumina-based or short read-based sequencing approaches. And the reason is that these organisms tend to have incredibly low GC content and stretches of AT repeats, which means that both the sequencing reaction and the subsequent assembly are incredibly challenging if you're relying on short read technologies. In fact, if you go to NCBI and you look at most Fusobacterium genomes, you'll see that most of the ones that are publicly available were sequenced through Illumina-based approaches, and that most of them are incomplete genomes. So the ability to use PacBio smart sequencing has been a huge advantage to us. And it's allowed us to obtain this complete and closed genome for this isolate. And this indicated to us that F. sphaericum has a 4 circular contig assembly. We actually sequenced this isolate multiple times because this 4 contig assembly is also incredibly rare for Fusobacterium species members, but it comes back as a 4 contig assembly every single time. So we really believe that it's real. And when we analyze the genetic contig across these contigs, we observe that F. sphaericum lacks all canonical or all known fusobacterium virulence factors. Now, because we have physical access to this isolate, we can take this isolate into the lab and ask, well, if F. sphaericum lacks all canonical virulence factors, does it still display any of the virulence phenotypes that we typically see with fusobacterium species members? And when we do that, we observe that F. sphaericum is still able to adhere to colonic epithelial cells and is still able to induce secretion of interleukin-8, which is a pro-inflammatory cytokine. So on the left here, confocal imaging of colonic epithelial cells, or HCT116 cells, co-cultured with either F. nucleatum or F. sphaericum. So the co-culturing of HCT116 with F. nucleatum is very well known in the literature, and it's known that this fusobacterium species member is able to adhere and invade these colonic epithelial cells. Sorry, I should mention in all of these images, the bacteria here are labeled in red. And so in the F. nucleatum case, you can see red both along the cell edges and then within the eukaryotic cells. In contrast to that, what we observe with F. sphaericum is that F. sphaericum is still able to adhere to these colonic epithelial cells, so we still see it tightly associated at those cell junctions, but it seems to be unable to invade or become intracellular. Despite that, when we measure quantitative levels of different chemokines and cytokines, we see in particular that there's still a measurable amount of interleukin-8 that is secreted by eukaryotic cells that are interacting with F. sphaericum. So here on the right, I'm showing you the full change on the y-axis, and you can see that while the levels of interleukin-8 for the positive control antibody cocktail or in the presence of F. nucleatum is higher, there's still a measurable amount of interleukin-8 in the presence of F. sphaericum, which is highlighted here in blue. Now I'm really showing you this data just as examples of the types of questions and the types of analysis that we can ask when we have physical access to these isolates. And this is why really having that physical access is so important to us. Despite the fact that traditional culturing can be quite successful, it has a couple of drawbacks, and the biggest of which is simply the sheer amount of time and people power that it can take to isolate these individual strains. So it takes a measurable amount of Petri plates and just time and people to pick individual colonies and grow them up, streak them out and grow them up over and over and over again until you get to pure clonal populations. And because of that, you are inherently decreasing and limiting your sampling potential. In order to address this and bypass this and exponentially increase our sampling potential, we turned to a large-scale culturomics approach. So in this approach, individual bacterial cells are grown up in individual wells. So this grid that you see here, each of those individual wells would be a well that bacteria are grown up in. And the individual cell is allowed to divide and grow into a clonal population. And that clonal population is then sequenced through Illumina-based approaches. So at the end of the day, you get two things. You get both your physical isolate because they've grown up in that well, and you also receive an Illumina-based genome to at least do preliminary analyses on. When we take this approach by 16S RNA sequencing, we noted that we could identify over 1,300 isolates. And when we blast those 16S rRNA genes and establish a 98.5% identity threshold, we see that 99 of these are of potential interest and potential novelty. And so we further Illumina sequenced those 99 isolates. I'm going to be telling you about 6 of those today, which had a novel 16S rRNA gene, and they showed a closest genome number of a species called Peptinophilus harrii. Because we had physical access to these isolates, we were able to apply PacBioSmart sequencing and obtain complete and closed higher quality genomes for all of these isolates for comparative genomics. And when we do that, we observe that our isolate does indeed have the closest neighbor of Peptinophilus harrii. So on the left here, I'm showing you the same type of phylogeny, a case-knit phylogeny, as what I've shown you before, but in this time, we're looking at Peptinophilus species member genomes. Our 6 isolates of interest are in magenta at the bottom, and all of the turquoise genomes labeled above it are genomes that are currently annotated as Peptinophilus harrii in the NCBI database. However, what we noted is that our 6 isolates of interest actually group away from the Peptinophilius harrii type strain, which is highlighted here with this arrow, and instead group away from it, suggesting and supporting the claim that our isolates represent a novel Peptinophilius species. Furthermore, we noted that 3 genomes that are currently annotated as Peptinophilius harrii, here highlighted with this dashed box, also group away from the Peptinophilius harrii type strain, and instead group closer to our isolates of interest, which suggests that these 3 genomes might currently be misannotated as Peptinophilius harrii in the database and are actually, in fact, the novel Peptinophilius species that we've isolated. Another way that we can define bacterial species is through a metric called average nucleotide identity, or ANI, where there's an established 95% species threshold. So what this means is 2 genomes that have an ANI greater than or equal to 95% are considered to be the same species, while 2 genomes that have an ANI of less than 95% are considered to be distinct species. When we calculate this ANI metric between our isolates of interest and the Peptinophilius harrii type strain, we indeed see that the ANI is below 95%, confirming that our isolates represent a novel Peptinophilius species. Furthermore, when we compare our isolates of interest to the 3 genomes that I've highlighted in that dashed black box, we see that the ANI is greater than 95%, supporting the claim that these genomes are actually currently misannotated and are indeed representatives of the novel Peptinophilius species that we've isolated. Because we have really high quality genomes, we can go one step further and start looking at how gene content from our isolates might differ to other known species. So we can use tools like this Genes in Genomes, or GigMap, which was developed by Dr. Sam Minot here at Fred Hutch, and very quickly look at gene content. So in this visualization, each row represents protein coding genes in that genome, where blue represents presence and white represents absence. So I essentially think of each of these rows as the protein coding barcode for each of these genomes. You can very quickly then compare down a column to look at how gene content differs from one genome to the next. And when we do this, we can detect that there is gene content that is present in our isolates of interest that is absent in every other currently available Peptinophilius genome. We can go one step further and start comparing between our isolates and look at the predictive functions of gene content that might be specific or unique to one or the other to start informing about the biology that these may be capable of. So as an example, we observed that there was gene content that was present in this top isolate called 7 that was absent in this bottom isolate called GALT25. And most of this gene content was present in a plasmid that was harbored by GALT7 that was absent from GALT25. One of these genes was of interest to us because it's an Ig-like domain-containing protein, which is an ortholog to a Listeria monocytogenes internalin. And in Listeria, this internalin mediates bacterial adhesion to and invasion of eukaryotic cells. And so we can start asking questions and testing questions like, does the GALT7 strain have greater adhesion and invasion capabilities than the GALT25 strain? So I'm not going to show you that data today, but that's just an example of one of the interesting roads that this type of analysis and this type of approach can lead us down. So because our end goal is obtaining physical isolates, right, both traditional culturing and large-scale culturomics inherently depend on your ability to culture a bacteria. And just because a bacteria doesn't grow up doesn't mean that it's not culturable. It actually more likely means that you haven't found the right conditions to grow it in. And that's not an insignificant challenge. That is a huge hurdle to cross from one bacteria to the next. And so in order to bypass that hurdle, we decided to additionally apply a culture-independent single-cell whole genome sequencing approach. So I'm not going to go too much into the details of this approach, but essentially, individual bacterial cells are encapsulated. They're lysed within these capsules and their DNA is amplified. The capsules are then sorted for ones where DNA amplification was successful, and they are then sequenced through Illumina-based approaches. And it's that Illumina data that we work with. When we assemble that data, we get what we call single-cell amplified drop genomes, or SAGs. And we can identify the closest related public genome to each SAG via blast analysis. For those SAGs that don't seem to have a close neighbor or closely known neighbor, we can bioinformatically characterize them further with some of the tools that I've shown you today, as well as others, and start assessing at least what they might be close to. I'm going to tell you about one of these SAGs today, which is an isolate called IF-00034. And this SAG showed distinct content, with its closest neighbor hit being a species called Porphyromonas asaccharlytica. Now, because the approach we'd taken here was by design a culture-free approach, this approach in and of itself did not give us physical access to our isolate. But we were really lucky that we had also applied a traditional culturing approach, and we had actually isolated this exact same strain from that approach and had already applied PacBio Smart sequencing to obtain a complete and closed genome. When we compare that isolate genome to other known Porphyromonas through a case-net phylogeny, like I've shown you before, we do indeed see that IF-00034 has a close neighbor of Porphyromonas asaccharlytica, as well as another Porphyromonas species called Porphyromonas eunonis. When we compare the ANI of our isolate to other publicly available Porphyromonas asaccharlytica and Porphyromonas eunonis genomes, we see that the ANI is less than 95%, that 95% species threshold, which supports the claim that our isolate is a novel Porphyromonas species. So in conclusion, what I've shown you today is that traditional microbiology approaches can be really successful and lead to the identification of novel species. And I think the Fusobacterium sphaericum story is really a nice example of that. The drawback, however, is that traditional microbiology approaches can really be limited by bacterial growth and really just inherently time and sampling potential. So to overcome these limitations, we've applied a combination of large-scale culturomics and bacterial single-cell whole genome sequencing to 4 tumor tissue samples from patients with colorectal cancer, and we successfully identified 4 novel bacterial species across 3 of these patient samples. I've shown you a little bit about how we've used each of these techniques, and hopefully it's become apparent that these techniques come with their own sets of limitations. So no individual technique own is perfect, but if you can apply them in a complementary way, it can allow you to really deeply characterize tumor-associated bacteria. And I think this last example that I gave you with the novel Porphyromonas species is a really nice example of how we could use 2 different approaches to get down to this is an interesting road to go down. Now, application of various bioinformatic tools allows us to assess, species and strain label novelty and map microbial genes and functions that might contribute to tumor niche adaptation to further start understanding what the biology that is happening there might look like. So with that, I would like to thank all members of both the Johnston and the Bultmann Lab, specifically those that are highlighted here in blue, who have been involved in various of the projects that I've mentioned to you today. I also really want to thank Dr. Sam Minot here at Fred Hutch, whose computational knowledge and assistance I deeply respect and admire. And I'm constantly seeking out, not just in the projects I've shown you today, but in many of my other projects as well. I also, of course, want to acknowledge our funding sources and say thank you to cancer patients and their families for tissue donation, without which none of these studies would be possible. And with that, I would like to thank the organizers for the invitation to talk and you for your attention today.